Hello friends, a warm welcome to all of you in today's session. Today, myself Nalini Tiwari, a molecular taxonomist working on Earthworm Biology Laboratory, Department of Zoology, Dr. Hari Singh Gaur, Central University, Sagar, I will discuss here about identification of earthworms for vermicomposting. Today in this session, you will learn about the basic structure of earthworms and how to identify the earthworms for vermicomposting. Friends, as we know that earthworms are also known as farmer's friend because of their valuable contribution in soil fertility and structure. They play an important role in vermicomposting as their casts were used in organic farm as a compost. But friends, do you know that all earthworms are not useful for vermicomposting? So in today's session, we will learn how to identify earthworms for vermicomposting. Earthworm fauna of India is well reported in compared to other Asian countries. Presently, 435 valid species and subspecies were recorded from India of about 10 families and 71 genera. Out of them, 71% of genera and 89% of species were endemic to India. Endemism rate is very high in Indian earthworms. Only 45 species were reported exotic in Indian subcontinent. Out of this 10 families, only species of 4 families were used in vermicomposting, that is Moneligastridae, Udrilidae, Lumbricidae and the Megascolicidae. Let's start with basic structure of earthworm. Earthworm belongs to phylum Annelida, kingdom Animalia, is a terrestrial organism which lives in a burrows. On the basis of their habitat, feeding and burrowing habits, earthworms are grouped into 3 categories. That is, epigeic, upon the earth, they are litter and surface dwelling species. Endogeic, within the earth, they are soil dwelling or upper soil surface species. And the NSS, out of earth, they are deep burrowing species. Generally, the epigeic earthworms are used in vermicomposting. Now let's talk about the taxonomic parameters to identify earthworms. Among taxonomic parameters, the 3 broad categories were established, that is the external characters, the internal characters and the molecular identification. Among external characters for identification, we have to mainly focus on the length, the diameter, the number of segments and the body color of earthworm, the type of prostomium, the type of setal arrangement, the position of several apertures such as spermatical pores, the male gonopores, the female gonopores, the presence or absence of dorsal pores and the presence or absence of genital markings. Now let's explore the morphological features of earthworm. Earthworm have an elongated cylindrical body which shows bilateral symmetry. The earthworm body is divided into several segments. These segments were called as metameres. The first segment of the earthworm body is called the peristomium. The peristomium has a ventral opening called as mouth, to which is attached a lobe-like extension, the prostomium. The joint of peristomium and prostomium differs in different species and thus they play an important role in identification purpose. The different types of prostomium are prolobic, when a separating groove not extending to the first segment mark, it is called prolobic. Epilobic, when it extends more markedly on the first segment, called as epilobic. Tannilobic, if the tongue touches the groove between first and second segment, it is called tannilobic. And lastly, the gygolobic, if the prostomium is not marked off by a groove from the segment, it is called as gygolobic. Now you can see a dark color median vessel on the dorsal side of the body. This is called as dorsal blood vessels and usually this side of the body is dark colored pigmentation. In certain families, a series of pores or apertures are found in the middle of the body which is called as dorsal pore, on the basis of which we can identify the species. While on the ventral surface, the body color is light or pale in pigmentation, and it has several apertures such as male gonopore, female gonopore, and the spermatical pores, which plays an important role in identification purpose. Now we can identify a mature earthworm with a prominent band-like structure, which is usually found in segment 14 to 16, called as clitellum. It is of different shapes such as flared in Glyphidralis, saddle-shaped in Pontus collex, and annular in Eutyphius, Metaphor, and several species. On the basis of clitellum, the body parts of earthworm is divided into three different parts: the postclitellar, the preclitellar, and the clitellar region. Next parameter, the CT. All the body segments have a tiny bristle or a spine-like structure or hairy structure called as CT. It is a locomotory organ which helps in locomotion of earthworm. The number and position of CT varies in different species, on the basis of which we can identify different species of earthworms. According to their number and arrangements, they are of two types: lumbricine or pericetine. Lumbricine generally have 8 CT per segment in pairs on ventral and latroventral side, while pericetine have numerous CT and are arranged in a ring on the segment. Earthworms have several apertures such as pairs of spermatical aperture. It is situated on the ventral side of the body, and their number and position is a taxonomic characters which varies in different species. Another aperture present on the ventral side is female gonopore. Usually it is present on 14 segment, while in some on 13 segment. It is also a very important taxonomic parameters for identification of earthworm. Another aperture found on the ventral side of the body is the male genital pore. Maybe it is found on 17th or 18th segment or 10th or 11th segment, such as in Dravida. Numerous minute pores called nephridiopores open on the surface of the body. It is difficult to see these pores. A very unique alternate arrangement of nephridiopores are found in Paronyx sensibaricus. A view of alternate arrangement of nephridiopores in Paronyx sensibaricus. Here you can see the alternate arrangement, that is, in one segment we can easily see the nephridiopores, a minute pore-like structure, while on adjacent segment the pore is absent. Nephridiopores helps in the removal of waste material from the body. It was similar to human kidney, where kidney excrete urea while nephridia excrete ammonia. There are certain areas which are somewhat depressed or swollen on the ventral side of the body, called as genital marking. Their number and position differs in different species, and thus it plays a valuable taxonomic parameters for identification of earthworms. Further, in internal characterization, we have several features such as alimentary canal, position of pharynx, number of gizzard, number and position of calciferous gland, segment from which intestine begins, position of ceci, typhlosol, if present, number of lateral herds, position of last herd, type of nephridia, that is holonephridia or meronephridia, and their position and number of testes pair, funnel, seminal vesicle, spermathecae, ovaries, ovisac if any. Position and type of prostate gland, that is of tubular or racemose. But to identify earthworms up to family level, we only need to focus on prostate and the nephridia. That is, prostate of tubular is present in Octochididae family, while racemose prostate is mainly found in Megascolicidae family. Lastly, the cocoons. These are the cocoons. Depending upon species, they produce varying number of cocoons. These cocoons are deposited near moist soil surface and they vary in different species. Now let's discuss the identification key of earthworms of India up to family level. Indian earthworm families is subdivided by inconspicuous male pore and conspicuous male pore. Inconspicuous male pore, where male pore is not clearly visible, while in conspicuous male pore is clearly visible. The inconspicuous male pore is further divided into two categories, where in first, species having clitellum flared into wings, gygolobic and setae in regular rows throughout the body, that is of family Elmidae, while in next the clitellum not flared, irregular arrangement of setae, that is family Rhinodrillidae. For example, the Pontus Colex Corynthosaurus. While the conspicuous male pore is further divided into two categories, the species having male pores in front of segment 15, while others have the male pore behind segment 15. The species having male pores in front of segment 15 is further subdivided into two categories. The male pores at segment 13 or 15 in family Lumbricidae, while the species having male pores at intersegment groove of 10 and 11, that is of family Monelli-Gastridae. While on the other hand, male pores behind segment 15 is subdivided into two categories, species having dorsal pores and species without dorsal pores. The earthworm species without dorsal pores further subdivided into two categories. The earthworms having robust dark color, female pores large slit close to lateral margin of segment 14 and having male pore on segment 17, that is of family Udrilidae, while thin whitish worm having female pore tiny close to mid-ventral lines or at B lines on segment 14, that is of family Ochnodrillidae. While on the other hand, the species with dorsal pores is further subdivided into three categories. The families having prostate tubular and holonephric, that is Acanthodrillidae. The families having prostate tubular and meronephric nephridia, the Octochetidae, and lastly the prostate racemose and having meronephric nephridia, the Megascolicidae. Thus, by following this identification key, you can easily identify your earthworms up to family level. That's all for today. Hope you have enjoyed today's session. Now onwards, you will easily identify the earthworms for vermicomposting. Thank you.